Hello, I'm Julie Kornfield in Chemical Engineering at Caltech, and it's my pleasure today to share with you the work of two former graduate students, Karthik Ramachandran and Artemis Layanu, as well as a staff scientist, Tiziana Di Lucio, who will be giving a talk after mine that extends this work into materials nanocomposites containing tungsten disulfide to enhance the X-ray contrast of the devices and improve their strength. The project was initiated by Jim Oberhauser at Abbott Vascular, and we worked very closely with Mary Beth Kosuth, who's still one of the leading scientists developing these products. James has moved to Gilead, and we're also very grateful to Zhonghu Kai at Ariane National Labs, who helped us get started with X-ray microdiffraction, which you'll see in the talk. The motivation for the work is to enable a revolution in the treatment of coronary heart disease. Today, patients receive a metal implant, so a rigid permanent device, that opens the blood vessel, saving their life, but has some very serious long-term consequences, including requiring patients to be on anticoagulants for the rest of their life, and the inability of a rigid device to respond to increased demand. The blood vessel can no longer dilate, which can cause chest pain in the event that the person is more active. So an improvement on that might be a temporary device that holds the vessel open, allows the body to clear the plaque, and then disappears. And a remarkable thing about the device that Abbott Vascular developed is that the structural material is high L poly-L-lactide, which is a material that is known for being relatively brittle. And yet they created a device that has the ductility and strength required to tolerate crimping and deployment of the device, and created a morphology in the material that allows it to maintain its strength for more than 3 months in the body and then gradually dissolve. The advent of metal stents was a revolution of its own, saving many lives that would have been lost to heart disease. However, it has complications, including some long-term complications that are significant risks of death. So especially late stent thrombosis. So of the approximately 1 million patients in the US who receive a stent each year, approximately 50,000 will go on to develop late stent thrombosis, and of those, perhaps 12,000 will die. And that complication might be eliminated if the device were no longer there for a clot to form. And that has motivated decades of work dreaming of a device that could be temporarily present in the blood vessel. And among the various companies that are pioneering these kinds of devices, Abbott was the first to achieve CE mark approval and FDA approval. And then unfortunately, shortly after being approved, approximately 1 year later, the product was removed from market because it had a significant increase in the risk of major adverse cardiac events. And that provides the motivation for Tiziana's talk on a next generation of materials that would enable surgeons to better see the device and allow a thinner device to do the job that I'll tell you about today. The manufacturing of the polymeric scaffold superficially resembles that for metal stents, starting with the preparation of a thin-walled uniform tube, laser cutting the pattern of struts, coating it with a drug-eluting polymer, crimping it onto a balloon, packaging it, sterilizing it, shipping to surgeons, where it is then implanted in the patient and deployed. But the superficial resemblance is misleading because polymers dramatically change their morphology and properties due to processing. And in particular, I'll show you that the tube expansion step that Abbott developed for preparing the thin-walled tube and its interaction with the crimping process that they developed has some very non-intuitive effects that arise from developing tremendously different morphologies over a distance that is only tens of microns apart. It's a remarkable achievement that they made, and I hope you find the morphologies in the material as beautiful as I did as my students were discovering them. So we'll look first at this step of tube expansion, preparing a uniform thin-walled tube. The process would remind you of the way bottles are blown, where you have a glassy preform that, there it is, okay. A glassy preform that is heated, a pressure inside is imposed, and the material inflates until it meets the mold walls, which arrests further strain, allowing you to create a very precisely controlled, in that case of the bottle, a field of crystallinity and orientation. In the case of Abbott Vascular, they wanted to have a method that would allow them to get very uniform thickness of the walls, and blow molding is well suited to that, and minimize the duration that the PLLA is exposed to high temperature. So they came up with a process very much like blow molding. They prepare a preformed glassy PLLA tube, mounted in a device where they can heat it with infrared lamps, apply a pressure on the interior, inflate that cylindrical preform, and if desired, elongate it to impose a tailored combination of azimuthal elongation and axial elongation. So they refer to that as the tube expansion step. And then the materials rapidly cooled down and removed. And they discovered that the nature of the deformation history had a strong effect and a non-intuitive one. So down below, I show the two examples of deformation histories they imposed on the polymer. One is a 420 deformation. So it's a 400% strain, a fourfold elongation in the hoop direction, and a small, just 20% elongation in the axial direction, mainly just keep the tube straight. And then compare that to a 200 deformation, so a nearly equibiaxial deformation, which you would expect to give high strength. And as far as the tube, the expanded tube goes, that's true. So if you look at the properties of the expanded tube by using laser cutting to cut out a little dog bone along the hoop direction and to cut little dog bones along the axial direction, and you test them, indeed you find that the material that was exposed to a nearly equibiaxial deformation, the red bars in the bar graph, show much greater strain at break, and the ones that were exposed to a primarily unidirectional elongation in the hoop direction, they are weaker in both the hoop and axial directions. They're relatively brittle at the step of the expanded tube. When they looked at the scaffolds that are produced from each of the two tubes, the situation reversed. The 200 tube fractured dramatically. So in a test that mimics overdeployment, if the doctor inflates the device too far, they found that the devices would have sometimes uncountably many cracks because it would fall apart. And so the red bar that's labeled lower limit, that is when there were a countable number of cracks. And most of the devices had greater than 40 cracks in the struts of the device. Whereas the material that was subjected to the nearly uniaxial elongation during tube expansion, it gave a device that had very few cracks in this overdeployment test, and it had a peculiar morphology that developed, and it's shown here in an SEM image where you see what looks like a pit into the material at the inner bend. So if you had this initial boomerang-shaped strut in the laser cut device, and then you bend it, you create tension at the outer bend and compression on the inner bend. Then when you deploy it and open it up, it reveals that the devices that don't crack have this pit in them at almost every single inner bend. And similar morphologies are seen near the junctions that are nicknamed Y and W junctions. And so I'm focusing on the U-bends, but this is occurring throughout the device in regions where compression is imposed during crimping, and it opens up during deployment. You'll see these pits. I saw that, I thought that must be horrible. And they said, no, no, no. The devices that develop those don't fail. Okay, let's see if we can understand that. So the disconnect between the properties of the expanded tube and the crimped scaffold arise because of a dramatic change in material morphology and properties that occurs during the crimping step. And the material develops strong gradients of morphology. You can see here in the crimp device, you see evidence of fissures in the region where the pit will show when you open it upon deployment. You see strong orientation near the outer bend, particularly kind of in the middle of the thickness of the device. And I'll show you microdiffraction measurements that help us understand more deeply than you can with polarized light microscopy, where you see high birefringence. It could be oriented amorphous material. It could be oriented crystalline material. It can be a combination of the two. And so X-rays are really needed in order to understand what the structure is that was created during crimping. Prior to crimping, we characterized these materials, and the entire region is very uniform and has relatively modest level of crystallinity after the tube expansion step. I don't know if you can make them out, but see this line of little dots? At each of those points, we have diffraction data. I'm going to show you a small set of the diffraction patterns that tell the heart of the story of the morphology that develops during crimping and how it's key to the success of the device. So if you focus on one little journey, so imagine you're a little ant and you're walking microns at a time along a direction from the inner bend where we see the fissures and low birefringence to the outer bend where we see this evidence of strong orientation in the plane of this section. So it's in the theta Z plane. If you look at the diffraction patterns near the inner bend, you have relatively weak diffraction peaks, and they're very broadly misaligned, reoriented relative to their state in the expanded tube. And at the outer bend, the structure is enhanced in both the strength of crystallinity and the degree of orientation compared to the expanded tube. For comparison, what you see at this 100 micron point, it's kind of like a neutral point between compression and tension. That's similar to what you would see uniformly throughout the expanded tube. So the result is that the misorientation or reorientation of the PLLA leads to a morphology where I think of it as being like tectonic plates pushed together in the compression regions, reorienting out of plane, so that when you open up, you can simply pull those chains apart without breaking them. You don't see crazing and fibrils running across this void. It's as if the surfaces just cleanly and gently separate. And that in turn has implications for the load that's put on the outer bend during deployment. If there's a low force required to open the inner bend, then the outer bend does not get subjected to high compression. And its morphology is virtually unchanged before and after deployment. So this became very interesting in relation to hydrolysis, because we found that the X-ray patterns near the outer bend gave an explanation for another mysterious thing that Abbott had achieved, which is that their device during the first 3 to 6 months gains strength and ductility, even though it's undergoing hydrolysis. How does that work? And so if you look at specimens, and Abbott prepared long-term aged or long-term hydrolysis specimens that in academia we'd never be able to do it. So it's extending a grad student lifetime. So they had samples that had been subject to 9 months hydrolysis and 18 months hydrolysis, carefully storing the samples at the end of the hydrolysis period so that no further aging or degradation would occur. And then when we applied X-ray microdiffraction, we could see that the patterns at 0 months, so just after deployment, showed a very strong morphous halo. And that at 9 months, that halo at the outer bend is hardly changed. And so the structure with high crystallinity, high orientation is resisting hydrolysis for 9 months. And meanwhile, you can see the expected degradation of the less crystalline material at the inner bend, where the crystallites had been disrupted during the crimping process. And then at 18 months, you see that the hydrolysis is taking place throughout the inner to the outer bend, and ultimately allows the device to just disappear. So to summarize what we learned, the process that Abbott developed involves an interplay between different types of deformation in different sequence, and the interaction of the processing steps is key to the success of their beautiful device. The process of crimping creates dramatic gradients in morphology from the inner bend to the outer bend, and those gradients in morphology not only allow it to deploy gracefully and retain strength, but even allow the device to continue to have strength in the outer bend after 9 months of hydrolysis. And in fact, they were so good at developing a morphology that survives that they could even allow that to degrade much earlier, which is a benefit for subsequent devices that might be thinner. So the explanation for why the better expanded tube gives a terrible scaffold and vice versa, and the mysterious increase of strength and ductility with hydrolysis were all revealed in large part thanks to X-ray microdiffraction, allowing us to see the morphology that Abbott created. With that, I'd like to thank Abbott for funding the research and for all the in-kind support in sample preparation that made this work possible. And I'd like to thank Karthik and Artemis for their excellent work, and Zhonghu Kai for helping us get started in microdiffraction. And I'd like to acknowledge Tiziana Di Lucio one more time and recommend her talk, which is the one after mine. Thank you very much.